In this module, we'll be talking about what a genetic variant is. Knowing about genetic variants will help us with the details of genomic sequencing. That way, we can feel more comfortable with the bioinformatics and data science behind it. Except where otherwise indicated, this content is available for use under CC BY 4.0. This module has two main objectives. First, we will learn a little more about what we mean by genetic variant. Next, we will learn about the molecular structure of DNA. This will help us better understand the genetic differences we are interested in studying. We always want to start off science with questions. Many of these have to do with how we are made up from the building blocks of life, or how we are made up from the DNA code. Today we might ask, what is my genome sequence? How does your genome compare to my genome? How are we all unique? If any of these questions seem complicated to answer, don't worry. Most of these questions are pretty broad and could even lead to more questions than answers. There is still a lot that we don't know. Your DNA, along with your environment and experiences, shapes who you are. This includes how you look, such as your height, as well as your hair, eye, and skin color. For other organisms, it can indicate broad versus narrow appearance or small versus large features. DNA can also include the not-so-visible things in life, such as how old you might live to be, disease factors, or responses to certain drugs. For example, inheriting some genetic variants can increase your risk of diabetes or heart disease, or can reduce the effectiveness of some anti-seizure medications in people with epilepsy. A central goal of genomics is to map and understand the molecular basis for these relationships. We will talk about what a genetic variant is in a moment, but first let's clarify. What is genomics? How does the study of genomics relate to DNA and variants? The genome is just a fancy way of saying, all of the DNA. Genomes are specific to individuals, so your genome is all of your unique DNA. A copy of your whole DNA sequence can be found inside each of your cells. This means we can determine your genome from any one of your cells, such as the epithelial cells you collect in a cheek swab. Genomics is simply the study of genomes, of all of the DNA in a cell. You'll sometimes hear the terms genomic and genetic used together. While genomics refers to the study of all the DNA, genetics usually refers to studying a few genome positions at a time in greater detail. In other words, genetics is studying a subset of DNA while genomics is getting the big picture. Now that we feel more comfortable with the idea of genomics, what is a genetic variant? Genetic variants are small changes in the DNA somewhere inside the genome. These variants create a lot of the diversity we see in the natural world, such as different eye and skin colors or susceptibility to disease. Let's talk about some examples of variants. SARS coronavirus 2 has a genetic variant called the Delta variant. This variant surged during the fall of 2021. Changes in the Delta variant make it more infectious, while causing greater disease severity among people. There were not that many genetic changes to create the Delta variant. So why is its behavior so different? In the case of Delta, some changes occurred in the genome where it codes for the spike protein. The spike protein is what allows coronavirus to invade human cells. Here are a few more examples. Cavalier King Charles Spaniels have many genetic differences from their breeding history that make them look different from other dogs. Alongside those differences, they also inherit variants in some genes that can lead to certain conditions like mitral valve disease. Inheriting a harmful variant of BRCA1 and 2 genes can increase a woman's chance of developing breast cancer. For a woman with these variants, the risk of developing breast cancer sometime in her lifetime increases from a 13% chance to up to a 72% chance. These variants are linked with developing other cancers across men and women too, like ovarian cancer, pancreatic cancer, and prostate cancer. Knowing about these variants ahead of time informs people of their risks and helps them consider preventative action with their doctors in order to catch cancer early. Genetic variants in people are being discovered all the time. Because genomics looks across all of our DNA, it is playing a large role in detecting new risk factors for disease and designing new treatments that can change people's lives. We now know how important genetic variants are. But where do they actually come from? As mentioned previously, genomes are our entire molecular code made up of all of our DNA. When we zoom in, we see the letters A, C, G, and T over and over again. These 4 letters make up your entire genome sequence. It's pretty remarkable when you think about it, that we are built with a code that only uses 4 letters. But in a similar way, computers do incredible work just using zeros and ones. What do these A, C, G, and T's actually represent? When we zoom in to look at the chemistry, we see that the building blocks of DNA, or the molecules that make up the 4-letter code, are nucleotides. Each nucleotide consists of 3 key parts: the phosphate group, the sugar, and the nitrogen base. The nitrogen base can take on a couple of different shapes. It can be any one of 4 shapes that represent the 4 letters. The A, C, G, and T come from their chemical names: adenine, guanine, cytosine, and thymine. We know that the human genome is about 3 billion nucleotides long. In the genome, a nucleotide occurs about every 0.34 nanometers. So even though it would be super microscopic in width, your genome is about a meter long when totally stretched out. The genome size of different plants and animals actually varies quite a bit. The human genome is about a meter stretched out. The genome of the canopy plant, Paris japonica, would be 51 meters stretched out. This could be as tall as a 14-story building. Meanwhile, the genomes of bacteria can be very small. The genome from this bacterium from the Nasuya genus would only be 0.04 millimeters if stretched out. Virus genomes can be even smaller. For example, the hepatitis B genome is only about 3,000 bases long. This is only 0.001 millimeters when stretched out. One of the big mysteries of genomics is that we really don't know why there is so much variation in genome size. Check out genomesize.com if you are curious about the different genome sizes of different animals. How do all of those nucleotides get put together to make up a genome? Nucleotides are unique in that they are able to polymerize pretty easily with the help of enzymes. This makes the long chains of DNA that make up the genomes. In order to keep track of which direction the long DNA strands are growing, scientists have labeled the carbon atoms on the nucleotide. We refer to them as 1 through 5'. It turns out that free-floating nucleotides always attach their 5' carbon to an open 3' carbon on a growing chain. Just like people usually line up facing the same direction, DNA synthesis always occurs from 5' to 3'. Those chains of DNA get copied into RNA through a process called transcription. RNA is also made up of nucleotides, but they are slightly different in structure and the pieces of RNA are single-stranded instead of double-stranded. RNA is convenient for the cell because it acts as an intermediary between DNA and the next steps. Specifically, the strands of DNA stay safe while the RNA copies go out and do the hard work. You can imagine the DNA as the original copy of a construction plan for a house. RNA is like the scanned copy that is used on site or is given to other people. Next, the RNA is copied into a protein in a process called translation. Translation is more complicated than transcription. In translation, the nucleotides that make up the RNA have to be read and turned into a more complicated structure. This structure is a chain of amino acids that will eventually get folded into a protein. Instead of only 4 different bases to choose from for nucleotides, there are 20 different amino acids that can be found inside a protein. During translation, we look at 3 nucleotides at a time on the RNA. Those 3 nucleotides are then translated to an amino acid using the pictured key. This is just like using a key to decipher a secret code. For example, 3 G bases in a row mean an amino acid glycine is added to the protein. You can see how quickly you can get a lot of variety and complexity from just 4 nucleotide bases. As I said before, RNA is like the working copy of the house construction plan. The protein that results is like starting to see assembled pieces of the house come together. Going from DNA to RNA to protein is a very important concept for creating the complexity we see in life. This is also known as the central dogma of molecular biology because it is so important for linking the changes in DNA to the bigger things we can actually see, like hair or eye color or disease risk. Now that we are familiar with the structure of DNA, where do the variants in DNA actually come from? Sometimes there is a single mutation in DNA that is converted into RNA and passed along to the protein. These can sometimes be devastating if they are in an important gene, causing disease in people who inherit the variant. Genetic variants caused by a single nucleotide change are called single nucleotide variants, or SNVs. Sometimes many nucleotide changes in the DNA are required to produce a noticeable change. It's also possible to have many nucleotide changes that don't affect the final outcome. This is because not all of the DNA is copied into RNA. The three-letter code that converts RNA to amino acids also allows a little wiggle room for mistakes. For example, both GGG and GGT get converted to glycine. You can imagine this redundancy comes in handy when a mistake appears. In our house analogy, we might be able to use different brands of building material without changing the final outcome. Finally, let's talk about what a variant might look like in data form. DNA is double-stranded. This is really useful for a couple of reasons. First, it adds stability to the DNA strands. Once they are double-stranded, the strands are harder to break and change. This is important because lots of changes in the DNA could mean unexpected changes to the proteins. Second, being double-stranded makes DNA easy to copy. When DNA is being copied, the two strands split apart and both are used as a template. It's much easier to be accurate when you have a perfect example to copy from. Conveniently, we can figure out what the other strand of the DNA is by using the base pairing rules. This is why you might hear the term base pair to describe the A and T circled here. Because of the base pairing, data can be represented as a long series of A, C, G, and Ts without their complementary base pairs. The example shown here is part of the genome of coronavirus responsible for a surface protein. Genome data in this format is much easier for a computer program to compare. It allows us to quickly process millions of lines of bases to look for variants. In this module, we learned a little more about what we mean by genetic variant. We also learned about the molecular structure of DNA that makes up the variants. This helps us target the variation that we are interested in studying. Thank you to everyone who contributed.